Let me introduce our next speaker. So this engaging morning is about to continue with a leading figure in neuroscience and open science advocacy, Professor Björn Brembs. Professor Brembs is an internationally renowned neurobiologist whose research has significantly deepened our understanding on how brain generate behavior, learn, and adapt. As professor of neurogenetics at the University of Regensburg, he directs a research group focused on the mechanisms driving spontaneous behavior, operant learning, and decision-making using both invertebrate and vertebrate model organisms. He completed his PhD at Würzburg University with a pioneering work on learning in fruit flies and a postdoc in Houston investigating the neural basis of learning in aplysia. He received a prestigious Heisenberg Fellowship for groundbreaking studies on learning and memory and made several influential discoveries. Beyond experimental science, Professor Brembs is one of the most prominent voices for open science and scholarly communication reform, advocating for transparency, accessibility, and fairness in scientific publishing. Throughout his career, he has combined rigorous inquiry with an inspiring commitment to improve the practice and culture of science. His ongoing efforts continue to shape both neuroscientific and broader research communities. He refused to use the microphone and refused to stick in this position. So, having said that, please join me in warmly welcoming our second speaker, Professor Brembs.
All right, so does that change anything in any way for anybody?
No. Perfect. Just as
All right, okay. All right, good. Okay, with that out of the way, Simon's slide. So there's nothing wrong with Simon's slide. I'm not going to put him on the spot and saying there's something wrong here. That's, I wouldn't even know if there was anything wrong in any of his slides. But I think there's something important that Simon didn't emphasize. And of course, I would think that this is important because that's what I work on. But that doesn't necessarily mean that it is important, because when you work for something for many, many years, you can get a warped perspective in thinking, oh, the most important thing in the world is the stuff that I study. And not everybody usually agrees with that, because otherwise everybody would study that. So I leave it up to you to decide whether you also think this is an important point that's missing. And maybe we disagree. And what is missing here is that, for instance, in the chemical circuit that Simon outlined, the concept of, input and output connections, specific input and output connections that combines input and output in a way that the output is not like the input. And that Simon said, well, this is something that generalizes to a whole lot of circuits, whether they're chemical, electrical, engineered or evolved, whether there are neurons in there or not. And that, of course, is correct. But what he didn't mention is that E. coli, when it moves around in space via runs and tumbles, it can switch between runs and tumbles without having any input. It does that spontaneously. It can do that. It doesn't, it does it according to input, but it also doesn't require the input to do that. And the same is true, maybe even more so, for the pyloric circuit, which is a gastric mill that runs continuously in a crustacean digestive system, and is famous for having lots of pacemakers, meaning that it runs without any input, and that there are both the individual neurons and the whole circuit can run without needing any input at all. So if there is no input-output function, is it still a circuit? And what Simon did mention, I think that's something that is really important, and he placed due importance on that, is the kind of diversity that we can see on all scales and in all perspectives in neuroscience. So, and the next slide is something that I didn't know until last year when I had to prepare a lecture for it, exemplifying how important it is to combine research and teaching, and that in contrast to the impression that some of you, the wrong impression that some of you may have, that your teachers and professors always know everything, which of course we don't. Something I learned last year, for instance, is that you can have animals, those are all mammals, you can have animals like a capybara that has a large brain, 76 grams, compared to a capuchin monkey with only 50 grams, but have way, way less than half of the number of neurons than the capuchin monkey. So obviously there's something here that varies dramatically, and those are not the only examples, but those are the most easy to see examples in this slide. So neuronal size and number vary dramatically, and we don't really have a good understanding if there's any clear rule or adaptive value to these kinds of differences. Another dramatic example is that of the animal that apparently has the smallest neurons. This is Megaphragma microwasp. You can see it here in scale to a schematized pyramidal neuron. You can see, so one neuronal cell body is already larger than the head of this microwasp, and you see some brain structure here inside. So it's tiny, and it has the tiniest neurons that people know. In contrast, take one of the largest neurons that people know. This is the growth cone of an Aplysia R2 neuron. This is roughly to scale like this. So you can see lots of these animals fit into just the growth cone of a single neuron. And this R2 neuron here, this is not to scale, right? That's the cell body is gigantic, and you can fit a gazillion of these little brains from that animal into just one neuron from another animal. So huge diversity in size. If then you look at the molecular diversity, single cell RNA-seq from a whole mouse brain, and what you find is lots of classes, supertypes, and 5,300 different types of neurons, just by looking at their gene expression. And then, for instance, you can take the Drosophila connectome that was published last year, and you find, well, 140,000 neurons, roughly 50 million chemical synapses, 8,453 annotated cell types. So you can imagine that size diversity does a lot to change the conductances, or the passive properties of a neuron, the biophysical passive properties of a neuron. The expressed channels and conductances that you find in RNA-seq do a lot to change the physiological behavior. And then, of course, the anatomy. When you have 8,500 different types of neurons, you can get a glimpse and an idea that probably, if you just look closely enough, there probably are as many types of neurons as there are neurons eventually if you combine all of these different properties. And so it seems a little bit weird that if you look at review articles on the types of neurons that you can find, if you look at their physiological categories, you find something like this, where fast spiking, non-adapting fast spiking, adapting irregular spiking, and you get some examples of those neurons. And you seem like this is really woefully inadequate to describe the sheer number of possible physiological properties that these neurons may have. And then if you look at a textbook to see what our students, what you have learned when you were at that stage, it's even more reduced. It's like, well, you know, neurons consist of a cell body, a dendrite, and an axon. And if you hyperpolarize them, they do this. And if you depolarize them, they all go like that. And when they're over threshold, they fire with action potential frequency that's proportional to the input. And it seems like that's a very, very dramatic simplification. And I don't think anybody really knows. These neurons probably exist that you have here. This is not a real neuron, this is a computer neuron, right? These neurons probably exist, but nobody really knows how common they are. They may be the exception and not the rule. And yet everybody thinks this is how neurons operate. And so you can find those neurons. This is an example of sensory neurons, where you have on the logistic x-axis, you have some value of input. On the output here, you have the firing frequency, and you see that they fire quite deterministic and linearly. This is what you would want from an adult sensory neuron. It should not fire when there's nothing that it's tuned to is available in the environment, otherwise you would be hallucinating. So that makes a lot of sense. But it may be that this is really just a special property of a single sort of general functional type of neuron that doesn't really generalize to any of the other neurons that are probably more relevant to the kind of cognition that we think of maybe going on, even though we may not all agree on what cognition is. And I shall also share that with Simon. I don't really know what cognition is either. So let's look at something so let's stick with the sensory input on the sensory side, the external processing. This is what, you know, information processing is usually sort of considered in. You know, you get information from the outside, you process it inside, and then something comes out. And you can look at that from the entire animal, or, you know, go down into circuits and single neurons. So let's stay on the outside of the animal. I'll go through this very quickly because this is all old stuff that most of you should be familiar with. So I just briefly remind you of Pavlov's dog. And so the input is either a conditioned stimulus like the bell, where the dog initially doesn't really care about, whereas if the dog, he or she is presented with food, it will salivate to it. And then, of course, as you're all familiar with, you pair those things together. The dog will salivate to the food and also register that the bell is associated with it. And then, in the end, the input of the bell is sufficient to elicit the salivation, the conditioned response. And Pavlov already realized the analogy here that it looks like one stimulus can supplant or replace the other stimulus. The bell can replace the food to elicit the response, sort of suggesting there's something peripheral, something sensory happening here, such that the stimuli are the important thing that are being learned here. And most famously, lots of animals have contributed to finding out how that works, except dogs. In this case, flies. Flies can also be classically conditioned. You put them in a tube here with an electrifiable grid, for instance. At odor B, nothing happens. With odor A, they get electric shocks. And when you then put them down here, where they can choose between odor A and between odor B, they usually choose the one that's not associated with electric shock. You can do the same thing with sugar. Also works, only there they prefer the one that was previously associated with the sugar. And already in the 1970s, as you can see here, mutants were discovered that are deficient in this type of classical conditioning. And Duns, the first one that was recognized, is phosphodiesterase. And rutabaga, another one that was discovered soon afterwards, is an adenylate cyclase. So apparently CAMP is something that was recognized very early on to be really important for this type of learning. And also in other animals, it turned out around that time that this CAMP molecule had something to do that was relevant to this type of learning. In flies, again, the kind of genetic tools that started to be developed over the decades can be used to not only find out which genes are important, but also which neurons that express these genes are the ones where the learning takes place. One such experiment you can see here. There's a lot of bars. We're only going to look at the first ones here and disregard the others for the sake of time. So what you see down here is what kind of genetic components you have in that fly. That fly does not have a 𝗕𝗷 line, which is the one that specifies where the genetic construct of interest is being expressed. It does have the rutabaga gene, and it does not have an extra rutabaga copy, a rutabaga cDNA, that in the other animals here can be expressed in those cells that this 𝗕𝗷 line specifies. And so if you just have the rutabaga mutant, it shows about half the learning of the wild-type control animals. And if you take a driver line that's called 247 that expresses in a structure that's called the mushroom bodies, and the mushroom bodies are an olfactory processing center, that's one of the reasons why they picked this particular line. And it when it does not express the rutabaga cDNA but does have the mutation everywhere in the body, it shows the same low learning score. But then if you express the rutabaga cDNA only in the mushroom bodies and everywhere else, the rutabaga gene is missing, it's sufficient to rescue the learning. Meaning that, okay, it's really, this is where the rutabaga gene is needed, so probably this is where at least some component of the memory is being formed. So that's what people have found out in, with these kinds of experiments. And to show that the mushroom bodies that are important for olfactory learning are not required for many other kinds of learning. Here's another fly experiment with a different set of stimuli. In this case, the animals are presented with upright teeth and inverted teeth. You can do this experiment in two different ways. You can just rotate those patterns around the fly, and whenever, let's say, the upright T is in front of the fly, you give it a punishing heat beam, and when the inverted T is in front of the fly, you switch the light off, or the infrared heat beam off, and which is a reward, so to say, because the animal is no longer heated, and you have ambient temperature. That's one way of doing it. That would be a classical conditioning experiment. You can also, by virtue of this torque meter where the fly is suspended that measures the attempts of the fly to turn left or to turn right, you can turn this into a flight simulator situation such that whenever the fly attempts to turn left, you rotate the environment to the right, so it gets a visual impression of actually having turned to the left. So you fool the fly into thinking that it can control its environment, but the only thing it can control is the angular position of those patterns. And then whenever the fly chooses to be in front of this T, you punish it, and whenever it chooses to be in front of that T, you reward it for having chosen the correct pattern. And afterwards, in both examples, both the classical and the operant conditioning, this is an operant conditioning example, in both of those cases, you ask the fly afterwards, you switch the heat off completely, and you ask the fly, okay, which of the two patterns do you prefer? Which do you want to have fly towards? And then you get these sorts of results. Those are called performance indices. There are 0 when the fly has no preference, and this is an 18-minute long experiment where in the beginning there is no heat. The flies can just fly around any way they want. This is an operant experiment, an operant example. And so they don't have an initial preference for any of the two patterns, just like Pavlov's dog initially doesn't salivate to the bell. Then they avoid the heat quite a lot. So +1 would mean that in this 2-minute experience, 2-minute period, the flies would fly only towards the unpunished pattern for 2 minutes and never fly towards any of the other patterns. And they get very close. Then there is an intermediate test, and they get even better. And in the end, you have some value here that shows that the flies, wild-type flies, have a preference for the unpunished pattern, even if there's no heat at all that tells them, oh, this is actually the punished one. They can remember the patterns. And you can do the same experiment with the rutabaga gene to find out where this is required. So first you show, of course, that rutabaga does not have a preference, and it doesn't matter how you train the animals, if you let the animals explore the T patterns, or if you just show them the T patterns. The wild-type flies learn this just fine. They show the preference, whereas the rutabaga flies do not show that preference. So it means that irrespective of whether this is operant or classical, the flies need rutabaga to learn. The difference between operant and classical, we'll get back to that. There actually is a difference between operant and classical, but it's more subtle than that. And then one of the things that, or the lines that people tested were central complex lines, meaning that they're deep inside of the brain. And the central brain itself is the area between the optic lobes that are cut out here. This is the central brain, and you see here the fan-shaped body, and for obvious reasons, you know why it's called a fan-shaped body, because it looks like that. And then you have 3 different 𝗕𝗏 lines that all express in this second from the top layer and also in other areas of the brain, but they overlap in this second layer. And they all rescue the pattern learning in the rutabaga mutant background. So it's the same strategy as with the mushroom bodies. You find, okay, it's this one particular layer. This is where the neurons sit that mediate the learning. And interestingly, this is pattern specific. So if you use the T patterns or something like this, where the vertical bars that are identical in both cases are just omitted, but it's the same, center of gravity is different, then this area up here, this layer up here, is the one that rescues. But for these patterns, it does not rescue. And for the layer down here, rescues these patterns, but not the other 2. And so what you can see here is that, okay, this is pattern specific, this is input specific. So much like the mushroom bodies are input specific to olfactory cues, these different layers are input specific to the different patterns that they receive, to the different visual patterns. So apparently, one rule that you can take from that is, from one, is that as I mentioned before, other animals have shown similar dependence on the CAMP cascade, the protein kinase A, and then for long-term memory, CREB, transcription, translation, and so on. But they all depend on the rutabaga gene to make CAMP. And the other thing that you can draw from not only the fly experiment, but also from the mouse aplesia, mouse and aplesia experiment, and experiment in many other animals, that apparently when you process external stimuli in a way that lead to some sort of Pavlovian memory, it turns out that this processing modifies sensory pathways, the sensory pathways that are specific to the input. And they use CAMP and PKA. Right? So this is a very general observation across all the bilaterian branches that people have looked at. So now to the more central part, and that is, okay, this is sensory processing. Sensory processing in many ways is not, for some people, is not necessarily considered cognitive. Cognitive is what we do with that information. After the sensory input has been processed, we derive meaning and we extract parameters, we do something in our heads. And so how do we become, as Konstantin said, how do we become who we are? How do we distinguish the internal from the external? Is that even necessary? So how do we distinguish self from non-self? I think that is probably very important. And Konstantin also mentioned the concept of reafference. We treat reafferent stimuli dramatically different, and not just we, all flies as well, treat reafferent stimuli dramatically different from the way we treat exafferent stimuli. And so all of this, I think, is important and eventually lead, in humans at least, to something that we call subjective experience, which is difficult to know if anybody else besides us has subjective experience. And by us, I mean me, because I don't know if you have a subjective experience, just as you can't know if anybody other than you has a subjective experience. And so one of the first discoveries, I think, that shows that there's more to animal behavior than just sensory input and then doing something with it. One of the first experimental, really obvious experimental results that have had an impact was the discovery of endogenously active neurons in aplesia by Barbara Alving in the 1960s that showed that not only can you have constantly firing neurons that don't require any input, but also you can get bursting behavior in various neurons. And that was something very new at the time, because people at the time really thought that's what neurons do. They sit still, they don't do anything until input comes in. So finding neurons that behave opposite to that was a revelation in many ways. And now there are other neurons. Very recently, 𝗕𝗏ö𝗿, also in aplesia, showed a neuron that can trigger behavior. And that neuron, actually, you give it a tonic depolarization, and it fires very, very arrhythmically. So it's not strictly random, and it's not deterministic as though bursting neurons that you can see that are highly regular. It's something in between, and what he discovered was that there's a nonlinear process inside, a molecular process inside the cell that seems evolved, and I cannot go into detail, but that seems to be evolved to provide that sort of variable firing, because what comes after that is a variable behavior, and that variable behavior the animal uses to try out different options in order to solve a problem. And so also here in this is an isolated nervous system of the leech, this is a leech, and put electrodes on it in the dish, and it just starts swimming. The patterns you can see in here are swimming patterns, and then it stops. And it starts again, and after a different amount of time, it stops swimming. My microphone doesn't work anymore? Oh, it stopped working, so the battery may be empty. All right. Okay, well then, I'll use this. So, because there's nothing that we can tie specifically, for instance, to this instance, when the animal was turning left for 2-3 minutes, 1-2 minutes, and then started turning right. There's nothing in the environment that we can pinpoint that really changed here. So these are actions, not responses. These come from the inside of the animal, just as we've seen neurons being able to do this, nervous systems being able to do this, so animals can do that too. And one of the things now that we can do, we can use that output, and we can do the same thing that Pavlov did with his animals. We can make arbitrary stimuli contingent on it, so maybe heat. That's an experiment that was designed by Martin Heisenberg, my thesis advisor, many years ago, long before I started getting into this. And in this experiment, let's say the animal is attempting right turns. The torque meter records this and then switches off the laser diode that provides the heat. And whenever the animal is turning or is attempting left turns, we switch the heat on. So now there's no stimulus there that tells the animal, okay, this is punished, this is not punished. So we can look at, okay, this internal aspect, this internal processing, the only thing that the animal now can do is, okay, I need to find out what it is that I just did. My antennae, was it my abdomen, my legs, or what was it that is switching the heat on and off? It has to do that internally. There's nothing in the environment that tells the animal to do that. It cannot see the heat. It's not directional. It's not that that the heat comes from the left or comes from the right. It just gets hot all of a sudden in an environment that's completely white. And that is, of course, an operant conditioning situation because the animal controls the one single feedback that we made contingent on its behavior. But of course, we need to compare this situation to the situation where there are external inputs, because as we've seen with the T patterns
seem to be requiring rutabaga no matter what, no matter how the patterns were learned. And so to control for that and to make the situation as similar as possible, such that the only thing that's really different between the two experimental situations is the thing that the animal is the information that the animal has available. Does the information come from the outside or does it come from the inside? And so we add colors to this experiment such that when the animal is turning to the left and is being heated, everything's green. And as soon as the animal passes the front line, so to say, and tries to switch to the right, tries to switch its direction to the right, everything comes blue and the heat comes off. So now the animal can learn two things. It can learn that its behavior, its own behavior, is correlated with the heat, and it can learn that the colors are correlated with the heat, just as before the patterns were correlated with the heat and you needed rutabaga. And then we can do, in these two experiments, we can take rutabaga and other genes, switch them off and see, okay, is there any difference between the external processing of external information and processing of internal information? And then you get, again, we have those PIs, we do 8 minutes of training as before, test them for 2 minutes and ask, what preference do you have? And you find, again, as with the T patterns, rutabaga, when we have the colors here, rutabaga does not show a preference, wild type does have a preference. Here we express an inhibitory peptide that inhibits all protein kinases C. This was known from the literature that if you do that in classical conditioning or factory conditioning experiment, this has no effect. Also here, it has no effect. In this case, we use a heat shock promoter. We heat the flies for a short amount of time that expresses this inhibitory peptide. So in these animals, PKCs are inhibited. There's no heat shock here. Those are the brothers, the sisters in this case, of those flies. And they do not express PKCI, so their PKC genes are expressing normally. Now what happens when we take the world away and there's only the internal self left as a predictor of the heat? Then what we find is rutabaga mutant flies learn this just fine, just as wild-type flies, even better a little bit. And if you inhibit protein kinases, this learning is gone, whereas the control flies learn this just fine. So apparently, to flies, it makes a massive difference, at least biologically speaking. We can't ask the flies if they find it different, but keep in mind, if they had eyelids, in this experiment, they could close their eyes, not see the colors. It would be the exact same experiment as here. And yet, you see these really dramatic biological differences that if you use external stimuli, it seems to be all rutabaga, but if you use internal processing, it's a completely different pathway. What we know now, due to Andreas Ewiner, a recent graduate student in our lab, is that it is the protein kinase, the atypical protein kinase. So flies have 6 different protein kinases C, and the atypical is the one that is important if we express a CRISPR-Cas9 construct in 2 different sets of neurons. I will get back to which types of neurons they are. If we express a CRISPR construct against this atypical PKC, in both of these neuronal types, the flies don't learn anymore, whereas here, the control flies, they learn just fine. What you can also see here is that, of course, the way in which we display our results have changed over the years. When before you expressed means and standard errors and that was good enough, now we try to be as transparent with our results as we possibly can. We show that each individual animal, whether it was punished on the right or punished on the left, is shown here. You show not only the medians, but also the means. You show the quartiles, and you show the density of the results here on the right in these rain cloud plots. So you see how not only the techniques develop, but also the way you display your results develop over time. Now, of course, we were interested in what other genes are involved other than APKC. Nobody really knew what other genes may be involved. And a few years before we did experiments in flies, there was a publication that humans, this is my daughter 14 years ago, that humans learn speech. So the speech component of language, the motor component, they learn how to articulate using the FOXP2 gene. It was later found that also songbirds learn their song using the FOXP2 gene. And so I was wondering, well, wait a minute, the way the flies figure out how to switch off the heat is conceptually the same as humans and birds learn the language. They try out. Nobody can tell them, and nobody does tell them, how to move your tongue and your larynx and your syrinx when you're a bird to learn how to articulate and how to sing the bird song. You have to try it out. And so that may be the same kind of concept, but as Chomsky pointed out to Skinner when he made the same observation, just because it looks similar conceptually doesn't mean it has anything, there's any relation to it biologically. In contrast to Skinner at the time in the 1960s, we have here now the situation where we can test for any biological relationship that goes beyond just the conceptual similarity. Because we can take the one FOXP gene that the fly has and knock it out in two different ways. For one, we can use mutants, those are the flies shown here, or we can express an RNAi construct in all neurons against the FOXP gene. And out here are the control flies for the mutant flies, and out here are the genetic control flies for the transgenic flies. And what you find is the same as with the PKC experiments before. You find that with the colors, the flies, all of the flies learn just fine, whereas without the colors, there's a specific impairment in the FOXP mutant flies. Meaning the FOXP mutant flies, not only are they learning mutants, they're learning mutants specific for this type of operant learning that has only access to internal information, internally generated information. And that's also apparently, Skinner was right with his intuition that the way we learn language has some operant components, and apparently those operant components evolved when the last common ancestor was around, the Urbilaterian, the last common ancestor between flies and humans. So what we can say at this point is, if you look at the kind of composite operant learning that has a word learning component like the colors and a self-learning component like the behavior, like the turning, left and right turning behavior, they can be biologically separated into a rutabaga CAMP dependent, PKA dependent pathway for the world learning, the sensory learning, and for the motor learning. It apparently involves APKC and FOXP. And also that, if you look at the data that we have from humans, from birds, from mice now later as well, it also appears that the APKC, FOXP components are also conserved across the bilaterian branch, just like the sensory learning components are. A brief update, or a brief addendum on the type of neurons where we have to knock out APKC in order to reduce the effect, to impair the flies. We generated, after we discovered that FOXP was involved, we generated flies that express a GAL4 driver in the FOXP gene. And so we can use that FOXP driver to drive the CRISPR construct for APKC only in FOXP positive neurons. And that was one of the lines that was sufficient to abolish the self-learning, the operant learning in the flies. So apparently we deduce from that FOXP and APKC need to be expressed in the same neurons in order to mediate the self-learning. And if you look, and that's what Andy did in his thesis as well, if you look at the FOXP neurons in red and the APKC neurons in green, you see there's no yellow neurons. Well, it's hard to see for everybody, but if you have the stack in your computer, you can zoom in and see it. There's no yellow neurons in the brain, but there's plenty of yellow neurons in the ventral nerve cord, and you can even see yellow nerves coming out, yellow fibers coming out in the nerves that go to the wings. And so what we hypothesized, then what was our collaborator Carsten Duch in Mainz then found, if you look at the muscles that control the position of the wings during the wing beat, those are the ones that generate torque, you can find the fibers of the motor neurons to be yellow. So there are indeed motor neurons that express both FOXP and APKC. These motor neurons are involved in generating torque. And so apparently they are somehow modified. This is the hypothesis that we have. They are somehow modified to make the fly then later turn more to the unpunished direction than into the punished direction. So you could think of one side of the animal, the neurons are modified in a way that the wing beats less. And then the other side, they are modified in a way that the wing beats more. And then if I beat more with my left wing than with my right wing, I would do more right turns. Even if the signal that comes from the brain was says turn straight, you would turn right. And if that would be the case, we hypothesized, then if we ask the animal, go left and after training, and go right after training, we should see an asymmetry, because before the experiment, we always set the left turning and the right turning maximal response to be zero symmetrical, to be symmetrical. How does that look like? I won't have time to go into the different muscles that 𝗕𝗿ö𝗿 went into. You do this with optomotor experiments. You give the fly an environment. This is an old slide from my mentor, Martin Heisenberg. You give a fly at the torque meter 15 stripes, let's say, and you rotate them counterclockwise, and the animal performs left-turning torque because the stripes go towards the left side of the animal, and clockwise or right-turning rotation give positive torque. These are 20 wild-type flies. And this is what we do before each experiment to calibrate that the fly doesn't that our torque isn't set to a way that the fly already has a preference in the beginning. And we've divided the flies here now in the flies that after the optomotor adjustments, they will be punished on, those flies that will be punished on the left are plotted in yellow, and those that will be punished on the right are plotted in green. And you see they're very similar. When the movement is to the left, they produce left-turning torque, and then when it goes to the right, they produce right-turning torque. And so if now the motor neurons have been modified, and we know anatomically that this is a pathway, the optomotor pathway goes directly from the brain via descending neuron to those motor neurons. So it jumps ahead of everything that's in the ventral nerve cord. So the motor neurons are the only overlap between the optomotor system and the self-learning system as far as we know. Then what we find is that if we provide the animals with a left-turning stimulus, those that have been punished on the left are, so to say, reluctant to turn to the left. They show a reduced optomotor response, whereas the ones punished to the right show exactly the same optomotor response as before, and vice versa on the other side. When you provide the right turning torque, the punished flies are attenuated, whereas the left turning ones are not. So we take this as evidence that it's the motor component that's being modified. And so what we have now here is a symmetrical aspect, so to say, or an orthogonal aspect, where the internal processing, it modifies motor pathways using FOXP and APKC. So different genes, different neurons, depending on whether it's external or internal processing. So dramatic differences. And you could even see, before you knew any of this, you could perceive these to be conceptually orthogonal as well. You could see that the Pavlovian kind of stimulus response concept assumes a certain conceptual idea of how behavior is organized. And now we know that it's the CAMP-PKA pathway that modulates that. Whereas if you would see behavior primarily as an endogenously generated action with some stimuli that can modify it, you'll see there's on the other end of that concept, there is a FOXP-APKC pathway that mediates the processing and the modulation of those pathways. So stark differences whether internal or external processing happens, which to me suggests you cannot just study one particular group of neurons or one particular set of learning, one particular set of circuits, and then generalize from those principles everywhere else. You have to check if you can generalize or not. And sometimes you can. You can between different sensory modalities and between different sensory conditioning procedures. You can do that. They generalize. But as soon as you have an internal processing, you cannot generalize to those anymore, at least not in terms of genes and in terms of neurons. So finally, for the rest now, why would one even have two different kinds of learning? If they're already separated, right? So if the sensory processing happens on the sensory end, and then the motor processing happens on the motor end, they're already separated. Why separate them genetically? And that may be because one answer may be that when this evolved, you didn't have nervous systems big enough to separate all these processes in space. You may have to have evolved neurons that can do both. And there is some evidence in aplysia that there are neurons that have both pathways and that can separate internally those two pathways depending on what the task was that the slug was supposed to solve. Another answer to that question is because animals always need to balance the internal processing with the external processing. I need to do certain things, and I have a certain stimulus situation, and they don't always square. It's not always that I am hungry that my environment signals food. It's not always that when I seek a mate that there is one available. It may be that I get food signals when I'm looking for a mate, and I get mating signals when I'm looking for food. And so I need to somehow, if I want to act, I need to somehow reconcile these things. And there need to be processes in how I can find a way forward in a situation that is not totally unambiguous. And so one way of testing how these kind of negotiations go on, when you have those two modules, is to put them together and separate them and see how much, when you had them together, how much of the processes that could have happened when both internal and external processes are possible, how much of that is left when you separate the two. Which means that you can train the animals here for 8 minutes with colors and with their behavior, and then you take the colors away and you ask, well, did you close your eyelids and realize that turning left is punished? Or did you just look at the colors and realize, well, green is obviously punished, that's sufficient. And what you find is that, in fact, the latter is the case. Wild-type flies, after 8 minutes of training, do not show any turning preference. Conversely, this must mean, I would say, that if they have learned green is bad, they should be able to use a different behavior to avoid green in a totally different situation. And so we provide a situation like this by coming back to the idea by rotating the environment around the fly in this flight simulator kind of situation. So in this case, they need to fly straight. Right here, they need to turn. They need to turn into the other direction to switch the heat off and to get rid of green and to obtain blue. And here, they have to fly straight. So if they fly, let's say, towards these two stripes, they're all identical, these 4 stripes. If they fly towards these 2 stripes, everything's green. So that was the color that was punished before. They should avoid it. But they should, in order to avoid it, they have to fly straight towards those 2 stripes, any of those 2 stripes, and then everything is blue. So not turning anymore. The turning is just really brief. If it's green, quickly turn away, but then fly straight. If you keep turning left, as you've been conditioned to before, everything would just be green, blue, green, blue, green, blue all the time. So different behavior, different situation. And the flies can't do that. So I take this to mean that in this situation, the animals preferably learn about their environment. There's no need to change anything in my behavior if I know that green is bad. So strikingly and confusingly, this is an operant experiment, but the animals learn that green is bad about as independent of their behavior as if it would have been a classical conditioning experiment and they would have been prevented from interacting with the colors, because it's independent of the behavior. Now, it wasn't sufficient to see that there's nothing here, because without the colors, the 8 minutes are sufficient to train the animals. It's like, so why do the animals not learn? And so I thought, well, maybe they just need more training. So these were 8 minutes, and then we just doubled the training to 16 minutes and said, okay, now the flies have been turning left to avoid the heat for 16 minutes, maybe that's sufficient. And indeed, when we then take away the colors, the flies do prefer the unpunished turning direction. And in fact, when we then test them in this situation, they cannot avoid the color, the green color, with this totally different behavior. And what we didn't know at the time, this is quite some time ago, what we didn't know at the time is that the motor neurons are modified. So now, and I had an anecdotal observation that the turning movements in the unpunished direction were larger than in the punished direction when looking at these flies. I never quantified it. I thought, that was a weird observation. I had no idea what that would mean at the time. But now, this makes actual sense, because the motor system is so dramatically changed that the fly, perhaps, is more occupied with figuring out how to fly straight than it is with the colors that are happening somewhere in the background. And this happens in many other animals as well. It's called habit interference, when your motor system wants to do one thing and your cognitive system, so to say, I apologize, tells you to do something else. And it's called habit interference. Now, what we also wanted to know is this process that's happening with the colors that takes 16 minutes, is that even the same process as the one that takes only 8 minutes without the colors? And so we use the FOXP mutant flies and train them for 16 minutes, and they're impaired, meaning that this is also strong evidence that it's not just when the colors are there, there's a third learning mechanism that just takes 16 minutes. It's really that the same learning process that in one case takes 8 minutes to modify the motor neurons, in another case really takes 16 minutes to modify apparently the same motor neurons. And we have, that's one of, how that happens is one of the projects that we have going on in the lab at the time. And that project looks at the mushroom bodies again. Interestingly, even though the largest part of the mushroom bodies is concerned with olfactory processing, there's also, and this is the last few things that I'm going to say, the mushroom bodies also have non-olfactory inputs. And if we switch off the mushroom bodies, and even only certain subsections of the mushroom bodies, and then after 8 minutes of training, they look like flies that have been trained, like wild-type flies that have been trained for 16 minutes, meaning they show the habit, the premature habit in this case, and show the habit interference as well. So apparently, somehow the mushroom bodies may be sending an inhibitory signal that tells the motor neurons, no, don't get modified yet. Wait until we're really sure that this left turning thing is really something that we need to do. And so to find out how that signal arises, how the mushroom bodies find out, this is when you have to send the signal and what pathway it takes to the motor neurons. That's the project that we're currently starting on. So we have separate modules for external and internal processing, and they interact. I have shown you the interaction about that there is an inhibition, there's a facilitation that I didn't have a chance to talk about that we know next to nothing about. So there's a reciprocal interaction that makes sure that the animal always weighs the external and internal processing and finds out, okay, which is the one that I should prioritize, more the external or more the internal. And we're starting to find out the neurobiological mechanisms of those interactions. And what I think, and those are my last, you apologize, conceptual thoughts, if you allow me to generalize from flies more broadly to Bilaterians, as apparently, at least on the genetic evidence seem to point out that this is something that flies share with many other animals. The universe, as we know since quantum mechanics, so now this year exactly 100 years ago, the universe can be stochastic at times. We also know that there are deterministic components, otherwise we would not exist. We could not predict certain things, but most of the time it's always some sort of mix. We have a good idea what's going to happen, and even as scientists, we cannot, not all of our predictions are 100% flawless, and so most of the time we live in a world that is sort of underdetermined. It has stochastic components, it has deterministic components. Some aspects we can predict better, other aspects we cannot predict as well. And that's the world we evolved in. That's the work every bilaterian animal, and probably the animals before, every organism, every bacteria, every archaea, every plant has evolved in a world that has predictable and more or less unpredictable components, and a mix of really smooth mix of all that. Which means that what we can find is we can have neural mechanisms that can appear algorithmic, to an extent that they can generalize to circuits that we design, even though they evolved. But we also have components, and those are the ones I talked about, that are nonlinear, that can behave unpredictably, that can generate things, can generate actions on their own, that are there to deal with unpredictable situations. When I don't know what to do, I have to try out something. And so these neural mechanisms evolved, and they allow us to become autonomous. What these algorithms do is that they are passive. They respond to external stimuli. What makes us autonomous and makes us active, what makes us become independent from the environment, such that we're not slaves of our environment, that's what's happening on the right side. That's what's happening over here. And most of the time, both interact all the time. So you cannot easily separate them unless you have a genetic model organism and the right experimental tools that you can actually separate them. And so that's why I personally think what was missing, or what is missing in a lot of discussions on cognition and on how, on artificial intelligence and on how nervous systems in general operate. I think this is actually quite important. It may be that in large nervous systems, only very few neurons out of those many thousands of neuronal classes show the kind of nonlinear behavior that we would expect and to be, so far, we would expect to be necessary for that sort of activity, that initiating activity. Maybe that is only very few neurons. And so you could dismiss it as saying, this is just some noise component that we can just average out. I think that's a valuable position to take, and I think it's a valuable position to discuss. And always when you have this kind of gray scale, you will tend to people who emphasize this. You will tend to have people who emphasize this when most of the time you have some sort of mix. So I'm going to emphasize this and try and see where we end up. Thank you very much. The mic.
So I suppose it would be really dumb of me not to comment. But in fact, you've done me a great service, because I've missed something out in my treatment of circuits, and that is that I have these transfer functions which are deterministic, but in fact there are built into circuits elements that generate random behavior. And that the random behavior is very important for searching the world. And the stupid thing is I already knew that, because I taught bacteria chemotaxis, and in the E. coli circuit, it's turning randomly, but the probability of it turning is biased. And this bias is controlled by chemical reactions, and the randomness comes from the stochasticity of molecular binding, because very small numbers of molecules are concerned. And in fact, you can see the same things in neurons. Now, with respect to there being something else missing in my circuit, I looked at the E. coli circuit, and I realized actually that what was missing was reafferents. But it wasn't that you were talking about. What were you talking about was the ability to generate spontaneous activity that is in fact deterministic, as in pacemaker neurons and so on that are spontaneously active. And those do depend on a circuit. As you pointed out with the Plizia, they depend on an internal circuit. But anyway, you've done me a great favor. It'll come into my next treatment of this topic, and I will publicly acknowledge your contribution. Thank you very much.
Thank you very much. You're too kind.
Yes, over there.
Thank you for the talk. So I have quite a few questions, but I'll just ask one, otherwise then maybe we can discuss later. So how separated is this distinction of between the motor system and the sensory system? So let's say I would like to know what you think would happen in this completely hypothetical experiment. So let's say you've got two flies. One has been conditioned to avoid leftward, for example, direction and turning. The other one hasn't. Or maybe in the opposite direction, the other one. And then you remove the head of the fly, of one fly, put it on the body of the other one and vice versa. So will that actually cause the fly to reverse? I mean, not go with the body, not with the head. Is that what you're implying, or is it necessary that both components are?
That's an excellent question. We haven't done the experiments. Similar experiments have been done. Yes. I would expect that, so there's some technical hurdles. You have to get the flies to fly without a head, which is tricky. You can get them to walk without a head, fly. I'm not aware of people having tried that too much. Even already, if you have a dark environment, they stop flying in 10 seconds. But you put the head on. Oh, you swap the heads. You swap the heads.
I think somebody has done such crazy experiments. I don't remember if it flies or another, but I see it's unbelievable that somebody has done this stuff.
I think that can, so in flight that cannot work.
Exactly.
current interpretation would be, according to my current interpretation, it would be the body and not the head that would shift the envelope, so to say. So you may ask the question, why would the motor neurons change? And of course, there's proximate and there's ultimate reasons why you would do that. The ultimate reasons would be because we think we have tied into a system that the flies use when they have damaged wings. Right? So I think the system evolved to change the motor system such that the fly would just focus on flying straight, and the ventral nerve cord would take care of compensating for the damaged wing. So that's the ultimate. The proximate one, you could see that as it's sort of related, but the proximate one is that if you change the physiology of the output, what you do is you don't change the origin of the behavior, you change the envelope, and it's specific to a particular behavioral a particular behavioral output. So what that means is we still assume that the initiating activity comes from the brain, that shifts from left to right turning torque, right? That explores the different motor outputs to figure out what is it that turns on liquids. We think this comes from the brain. There's some evidence to show, there's some evidence that suggests it comes from the brain. It's not pure speculation. And if those would be the circuits that would be modified, it would modify a very central aspect of behavior. It may also affect other behaviors, such as walking to the left. But by just modifying the output, you leave the most important thing, being able to be active. You leave that intact. And only your behavior, so that before, the colors were considered hierarchically on top of the behavior. And apparently, the behavioral output is hierarchically on top, on the initiating aspect of the behavior, if you look at the behavioral output pattern. And so what you can imagine is you have like a layered set of protections that protects the animal from becoming inflexible. Right? Because as we showed, if you modify this behavior in a more permanent way, with what's the innate PKC in the motor neurons, it interferes with generalizing to other situations. And so that's why I think the situation, that we find what we find, that's an interpretation. And that's why I think if you swap the heads, the output will be dominated by the ventral nerve cord. Not to 100%. So the changing of the motor neuron does not explain the entire variability that we see in the fly's behavior. The flies can still, turn more to the punished side if the brain tells them to. But if you take unconditioned heads, they will just send the same output, and then the envelope will be slightly shifted, one to the right, one to the left, and you see a shift in the middle. It will be reduced to whether you had not swapped, but it should be that.
Hi, thanks a lot for the talk. It was really fascinating. I have first maybe a basic question, which is when the motor neurons get modified, is that what we sort of call muscle memory?
That's a good question. I'm not sure there's a good definition of muscle memory. It's more like an experience that you have when you haven't done something for a long time, and you say, oh, let's see if I can still do it, and then you can do it. I would say, from motor learning evidence in mammals, most likely what we call motor memory or muscle memory is distributed between cerebellum, striatum, and motor neurons, spinal cord, most likely. But we don't know for flies if that's the case. So I guess modified motor neurons is about as close as you can get to muscles. So it doesn't get any closer.
And then the next question, I hope I'm making the right links. So from what I understand, when you talk about your last slide with the neural mechanisms and you have algorithmic and nonlinear, right? Anyway, so would you say the algorithmic side is more like goal-oriented behavior, and then nonlinear would be something like, I don't know, like play or exploration?
I would say the parallel would be more like
one more question, sorry. So following up, then what you talked about so far is the external versus internal. So, but then what happens when you have, I guess, an internal feedback? So in terms of, let's say, something like locomotor play or dancing, then would you say the feedback is
We can take one final quick question by Professor Tommasi.
Thank you for your wonderful talk. Thinking about the internal versus external input, I see a problem of bootstrapping, like what comes first. And I wonder whether the argument that neurons start to fire even before synapses are formed during development isn't an even stronger argument to the idea of a sort of internal priority in this bootstrapping problem.
Okay, so before thanking Björn Brembs, just a final recommendation to be here sharp at 2:30 to resume the session. So thank you very much, Björn.